Hello and welcome to Principle of Cognitive Neuroscience Part A. As you may know, this course is divided in three parts. The first part being dedicated to main models, where I explain to you the vision of scientists on how the brain works according to their angle of observation. The second part is dedicated to main concepts, and those concepts characterize what define a result, a publishable result nowadays in cognitive neuroscience. And finally, all along these teachings that will be online, we will have a practical session on Zoom where I'll teach you how to manipulate the data, analyze it, how to write a paper, write a project, code, software, and what is a life in academia. And this will not be recorded, this last part, and it will be taught directly on Zoom through practical sessions. This teaching is coming with a conference which already happened, and you are very welcome to watch on our YouTube channel. It is a conference related to special issues that was published this year on the structural connectivity of the cerebral cortex. I think it would be a nice complement of this teaching. And every Monday morning from 9:30 and 10:30, you're also welcome to join us for the Neurochino, where we discuss directly like a journal club, but very informal, as exciting science of last week. So if you want to know what happened last week in science, just come and connect and get your update on what is exciting from last week. But without any further ado, I will start my new lectures on brain asymmetries. So brain asymmetries are all about looking at differences that are between the left and the right hemisphere in terms of anatomy and in terms of function. You may have seen this slide before. It's a slide where I explain the discovery of the language production center by Paul Broca, who discovered these regions after he's been seeing his former colleague Ernesto Burton reporting a patient who tried to shoot himself in the head, survived the accident or the suicide attempt, but the suicide attempt left him with an open wound on his skull. And during those, scientific meetings, Orbutin showed that if he was like putting a little pressure on the brain that you could see through the skull wound, the patient was not able to speak anymore. And this pressure was happening at the level of the inferior frontal gyrus in the brain. So Paul Broca found that fascinating, and by chance met with Monsieur Le Borgne, who was like a patient that was in an hospital, a psychiatric hospital, and developed a gangrene of the leg. And really why Le Borgne went to see Paul Broca was to have a surgery and get his legs severed to avoid the infection to spread and him to die. And Le Borgne, of course, came to see Broca and was not able to speak. The only word that he could say was "tan." And Broca saw that as an opportunity to get a direct access to his brain. So he gave him like some care, but like 2 weeks later, the patient died, and Broca got his body, extracted the brain, and reported this tiny lesion in the posterior part of the frontal lobe of Le Borgne. And by doing so, he was able to demonstrate for the first time the relationship between this lesion and the ability to speak. But a longest carrier, you need to understand at that time, you know, in order to do what we call clinical anatomical correlation, you know, you had to wait for patient to die to extract their brain to see what's going on. So that was a small n, and by chance, Broca was able, the longest carrier, to collect several other cases. I think the longest carrier managed to collect about 7. And in 1863, Eve writes that for the people who were not able to speak, the lesion was always in the posterior third of the third frontal convolution, which is the inferior frontal gyrus. This number seems to me to be sufficient to give strong assumptions. And the most remarkable thing is that in all the patients, the lesion was on the left side. I do not dare to draw a conclusion from this. I wait from new facts, but this is probably one of the first depiction of a function that will be lateralized in one hemisphere. And here, of course, we're talking about the production of language. Then after that, work on the lateralization of function was not really carried on seriously until neurosurgeons started to do this radical new strategy, and it was starting like in the 50s or 60s, when patients were showing like a severe epilepsy. Instead of removing part of their brain, they decided to cut the corpus callosum in order to prevent the epileptic seizure to spread onto the entire brain. And so by cutting the corpus callosum, the epileptic seizure was staying just in one hemisphere, reducing the overall impact of the seizure. But that led to some strange and interesting syndromes. And I have a video right after that that will tell you more about it.
I know the left hemisphere and right hemisphere now are working independent of each other, but you don't notice it. Now, you just kind of adapt to it. It doesn't, you don't have any feeling, it doesn't feel any different than it did before.
7 years ago, Joe had brain surgery to allay the effects of severe epilepsy. His surgeon cut the nerve fibers connecting his left hemisphere with his right. While the operation was a complete success, Joe's unusual case offers an extraordinary insight into the machinery of mind. This fiber system, the corpus callosum, is located midway between the two hemispheres. When it was surgically severed in Joe's brain, the transmission of information between the two hemispheres was halted. Michael Gazzaniga.
What we can do is play tricks by putting information into his disconnected, mute, non-talking right hemisphere and watch it produce behaviors. And out of that, we can really see that there is, in fact, a reason to believe that there's all kinds of complex processes going on outside of his conscious awareness of his left half brain. Joe, I'm going to show you some things. I just want you to tell me what you see. And here we go. You ready? Look right at the dot.
Okay.
Right. Okay, you ready? Look right at the dot.
Grapes.
Good. When Joe focuses on a point, look right at the dot, everything to the right of the point goes to his left brain, the dominant hemisphere for language and speech. Look right at the dot. So we can see here that when we flash a word or a picture,
Tree.
Joe is easily able to name it. There we go.
See it.
Close your eyes and let your left hand do a little work here. Okay, what do you got there?
Pan.
Okay, very good. Now, when a word or a picture falls to the left of a fixation point, that information goes to his disconnected right half brain. And as we can see here, Joe is unable to name it. But Joe is able to draw the picture with his left hand, the left hand getting its major control from the right half brain. What'd you draw?
Okay.
What'd you see?
Wheel on one side. I don't know where I saw the other.
So even though he can't name it, his left hand is able to draw out a picture of the stimulus, of the picture or word that we presented to his right half brain. What did you see?
Saw a hammer.
So just close your eyes and draw with your left hand. Just let it go. That's nice. What's that?
Saw?
Yeah. What'd you see?
Hammer.
What'd you draw that for?
I don't know.
What we have with Joe is just a dramatic example of a neurologic case that really allows you this window into the non-conscious and how powerful non-conscious processes are at influencing our conscious self, our personal self. What Joe and patients like him, and there are many of them, teaches us is that the mind is made up of a constellation of independent, semi-independent agents, and that these agents, these processes can carry on a vast number of activities outside of our conscious awareness. Even though that goes on, there's some final stage or some final system, which I happen to think is in the left hemisphere, that pulls all of this information together into a theory. It has to generate a theory to explain all of these independent elements. And so, and that theory becomes our particular theory of ourself and of the world.
This is, of course, fascinating. So, did you understand what really happened? This patient has these two hemispheres of the brain disconnected, and when you display a text or a picture in the right visual field that project in the left hemisphere is able to say what it is, because the language production is in the same left hemisphere where the visual information was projected. When you present this text, or if you present an image in the left hemisphere, it projects in the right hemisphere, it won't be able to say what it is. But if you ask him to close his eyes and to draw with his left hand, the left hand that is commanded, like that is in control by the right hemisphere where those information was projected, the patient is able to draw what he saw. What's fascinating is that if you present at the same time two objects, one in the left hemisphere and one in the right hemisphere, you ask the patient to say what he saw, and he's going to report only what he saw in the right hemisphere because it projects in the left hemisphere where he has the language. And again, if he closes his eyes and you ask him to draw what he saw, he's going to draw what he saw in the left hemisphere without really understanding what is going on. And this is a window into understanding how part of consciousness work through the ability to verbalize things. And because he's not able to verbalize things, this part of the information that was in his right hemisphere remain unconscious to his verbalizing representation of the world, verbalized representation of the world. This phenomenon of a brain lateralisation and brain specialization for given functions according to the hemisphere led Rochesbury to get granted the Nobel Prize in 1981. And he did this little sketch for his for the day he got granted, and that's in his press release, where you have the summary of his career, where you have this brain where the corpus callosum has been disconnected, allowing him, by testing patients after patients, to identify a specialization of given function for the left hemisphere, such as the right hand touch, the vision in the right hemifield, reading and writing, main language center because of speech and comprehension, calculation, and yeah, well, right visual field projection, we talked about it already. While in the right hemisphere, it is more about the manipulation, the spatial perception of object, the part of the word comprehension, nonverbal concept formation, and left visual field projection. And those, those are really different functions that seems to be specialized in one hemisphere compared to the other in patients for which the two hemispheres have been disconnected. Now, the corpus callosum is an interesting structure, and if you look at it from the mid part of the brain, you're going to see it like project from posterior to anterior parts. And so like the anterior part is here coded in red and cyan, and this red and cyan corresponds to the minor forceps usually, and includes the rostrum and the genu. And it projects mostly to the orbital prefrontal and inferior prefrontal cortex. With the genu, you can have like some projection also onto the premotor cortex. It goes that far. And then you're going slightly more posteriorly, and you'll have the rostral body, where you have motor and premotor cortex, and then in 4, in pink, you'll have the motor and the somatosensory cortex. And as you're going further with 5, which is a posterior mid-body, you'll have mostly the somatosensory cortex and the rest of the parietal cortex. 6 is called the isthmus, and that's where you have the tapetum that's going to reach out the temporal lobe. And 7 is the splenium, where you will have most of the occipital connections. And according to the different part of the corpus callosum that you disconnect, you can have different form of neuropsychological manifestations, for instance, and that's mainly because neurosurgeons have been trying different form of disconnection in order to minimize post-surgery disorder. But if you were disconnecting the entire part of the corpus callosum, some patient ended up with what we call the alien hand syndrome, which is involuntary intermanual conflict resulting in one hand interfering with the correct action. So it's like your right hand is going to try to pull out something, and like typically, like you do the dishes, your right hand is going to try to open up a bottle of soap, and your left hand's going to remove the bottle of soap, and you have this kind of battle between your two hands. If you disconnect a little more posteriorly in the body of the corpus callosum, you will be able to observe in some patients the anarchic hand syndrome, which is a complex involuntary goal-directed movement of one hand. So it's usually your left hand is going to do really what it wants, and you have no control over it, and that's why it's called anarchic hand syndrome. If you go a little more posteriorly, you can have a left hand tactile anomia, that's mostly if you close your eyes and you put an object inside the hand, in the left hand of the patient, it will not be able to say what it is. And if you go a little more posteriorly in the body splenium, you'll have a left hand apraxia, which is the inability in the patient to execute goal-directed movement on imitation. So, right, when the imitation is provided on the right hemifield, or if you give a verbal command. So typically you ask patient to comb their hair with their left hand, and they are not able to do it. If you go a little further in the posterior part of the body, you can also observe a patient showing a left hand agraphia. So they're not able anymore to write with their left hand, which, you know, usually with our left hand, we're not really talented in writing if we're not left-handed. In that case, they're absolutely unable to do it. Same posterior body area, some patient will show hemispatial neglect, which I described in previous lectures. And these patients are not able to report any more events that happen in their left visual field. And then if you go again a little further, this time into the isthmus, almost the splenium, the posterior part of the corpus callosum, then patient will show a syndrome which is like main étrangère. So they do not recognize this hand as their own hand. They think it's a hand of somebody else, or they try to explain it this way. And this is particularly true when this hand is actually outside the visual field, outside the right visual field where they can verbalize. So it's, you know, mostly for the left hand. And if you bring it into the right visual field, they will say it's theirs. Then you have a phenomenon of Dicatic listening suppression. And so like that's essentially when you present auditory stimuli simultaneously to both ears, the patient find easier to recognize verbal linguistic stimuli presented to the right ear and they tend to ignore those presented in the left ear. Then you also have, like that, still going a little further, bilateral cross-optic taxia. So, you know, if you asking a patient to reach something in the opposite side of their hand, so like you try to reach something with your right hand on the left side, they will systematically miss like the coordinate of that thing that they gotta catch. And that's also, you know, when you have a disconnection of the posterior body of the corpus callosum. Then as you're going further, you can have deficit in binocular stereopsis and versions. So that's a reduced ability to perceive three-dimensional object. And you're not able to do convergent or divergent eye movement while fixing target. So it is typically tested. So first, we only see depth and 3D because our brain is able to reconstruct the vision from our two eyes. And that's what you manipulate when we want to do 3D pictures or 3D movies. You have to give two angle of vision to your eyes, otherwise you'll see it flat, like when you're watching a normal TV. And that's also why, you know, with the Oculus Rift, they give you two separate information for the two eyes to give you the feeling that you are in a three-dimensional world. So like the patient with a disconnection at this level lose this ability to reconstruct this 3D representation. And because they lose this ability to reconstruct this 3D representation, they lose the ability to coordinate their eyes for fixation. So if you take your finger, I'm gonna try to show you here on the video, and you ask them to fixate your finger, and you're going closer to their nose and more distant to their nose, you'll see that they do not coordinate correctly the way they're gonna converge or diverge their eyes to maintain a correct fixation on this three-dimensional object being your finger. So this is if you disconnect the splenium. Then you also have signs of optic aphasia, where in the posterior part of the corpus callosum, if you disconnect the splenium, where the patient will not be able to name object presented visually, but are still able to name this object if you give them a definition of the object. So typically you show comb and you ask people to say what it is, they're not able to do it. But if you ask them to tell you what is the subject that you use every morning to coif your hair, they'll say a comb. Same thing in the splenium, you can have coloroanomia, which is not an achromatopsia, so they're still able to discriminate color and say that they are different, but they will not be able to name visually the colors that are presented. Then, if you go a little further, you have alexia without agraphia, which we typically call pure alexia, that has been reported for a disconnection of the posterior part of the corpus callosum. And so, you know, the patient are not able to write anymore, but they are able to read. And finally, with the disconnection of the splenium, you can have a left visual hemianomia and hemialexia, which will be the inability to name object presented in the left visual field. And the right hemisphere continue to maintain the capacity for visual recognition, but it has lost the access to contralateral language region, and that's why you cannot name those objects. And the same for reading. So if this is presented as the left hemifield, the patient is not able to read anymore, but in the right hemifield, he will be able, because it will project on the hemisphere that is able to speak. And this is for the behavior you can look at, and in terms of strength of connections between homotopic area in the left and the right hemisphere, we're going to have to turn ourselves to a method which is resting state functional connectivity. I might have showed to you this wonderful slide from my friend, Professor Margulis, where you can see the fluctuation of the board signal in the brain at rest. You can see how some areas seems to be synchronized in their fluctuation. In some, they share the same colors. And if they fluctuate at the same rate with the same colors, they're highly correlated. And scientists consider them as functionally connected. And by doing so, you can really assess how much areas in the left hemisphere are functionally correlated with areas in the right hemisphere, and report a graph of strengths of correlations. What's amazing with this connectivity is that the profile of connectivity that individuals can have is actually representative of the pattern, the individual pattern of activation. So if you know how the brain is connected, you can depict how it will be activated in terms of task-related functional MRI. So it's really putting forward the importance of connections and connection diagrams into understanding the functioning of the human brain. But if you apply this to the inter-hemispheric connectivity, you can do those beautiful figures that have been made by Marc Joliot of the mapping of the homotopic functional connectivity. And you can see how brain areas that are showing, that are on the A, at the A level, brain areas that are really red are showing very strong connections with areas in the control, with the equivalent areas in the other hemisphere. When they're blue, they have moderate connection with the same area in the opposite hemisphere. And green is also a quite strong relationship. And the bottom row is actually showing you how this strength of connection between the two hemispheres can be variable across participants. And you see, like it's some areas can be variable, mostly ventral areas, but the rest of the brain is quite replicable in terms of the way it is connected between the left and the right hemisphere. Now, I know what you're going to ask. What happens if we cut the corpus callosum to these correlations? And I'm glad you asked, because in 2013, O'Reilly has been trying this experiment in monkeys, doing a section of the corpus callosum and the entire commissure. So like no communication between the left and the right hemisphere. And you can see and that will be on the left part of this slide, that before the surgery in A, you have here a correlation matrix. And those are like all different regions and how they are interconnected. So if you look at the square on the left and you divide this square on the left in 4 separate squares, you can see that the left hemisphere is very well connected with itself, and that will be the top left square, and very well connected with the right hemisphere, that will be the bottom left square of this big square on the left. After the surgery, what we can see is that the left hemisphere is still very well connected with himself, but it is very badly connected with the right hemisphere, as we should expect. And the right hemisphere is still very well connected with himself. So, you know, that's really good, and that demonstrates that it works in monkeys. And we had to wait 4 years to have the opportunity to measure the same thing in humans. The patients that were showing like very strong epilepsy, and the neurosurgeon decided to come back to this old method of separation of the two hemispheres. You can see on the right panel of this slide, the experiment from Roland et al. In 2017, where before the surgery, you have a pattern of intra and interhemispheric connection that is well distributed and completely lost after the surgery, which will be the two connectivity matrix in the middle of the page. And like on the right part of the slide, you can see the difference between before and after. You can clearly see that you have clear differences that started to appear in the interhemispheric exchange between the left and the right part of the brain. So in sum, what we've been showing here is that the exchange between the left and the right hemisphere can be cut. And this stops the interaction between the hemispheres. There is no more correlation in the activity. This allowed to see that you have a dominance, and as soon as you do the surgery, you have a clear dominance that is visible for some function in the left hemisphere, such as, for example, language, and function for the right hemisphere, such as the visual representation and drawings and all these kind of things. However, what we also seen is if you cut the corpus callosum, the brain, like the patient behavior is changing radically, and you start showing syndromes that represent the challenge that your brain is having by not having this interaction between the two hemispheres anymore. And that would suggest that it's not a clear cut of function between the left and the right hemisphere. The two hemispheres have to work together in order to optimize your interaction with the environment. And this is just the top of what we've been able to observe in this patient. I'm pretty sure they have also other problems in their everyday lives that have not been reported. So don't take Roger Sperry Nobel Prize for granted. Functions are not ultimately completely localized in one hemisphere and the other. It is way more subtle than this. Way more refined than this. It's more a balance between the two hemispheres, with core, hub, and center being in the right or the left hemisphere. This is clear for all of you. Now, since fMRI has been so good to show change in connectivity when you severe the corpus callosum, how about using task fMRI to try to look in the population whether everybody is having the language located in the left hemisphere. And this is a beautiful work of Bernard Mazoyer, who's been able to show, and that will be the top row here, that if you take like 200 right-handed participants and you ask them to do a task of verbal production, you will have a pattern of activation mostly involving, and that's the two brains that you see on the right, the left hemisphere, and very little involvement of the right hemisphere. And it's been able to produce a lateralisation index. And if you look at the distribution of the lateralisation index for the green chart on the top, you see that most people are right lateralized for their pattern of activation. And you have a second peak of less participants that are a little less right lateralized, and like very tails that will be on participants that are showing weak or no lateralization. And then if you take 200 other participants and you take them and you're very careful that they're very strongly left-handed, and you'll see that those left-handed participants do not have systematically the language symmetrically organized compared to right-handed, like we might think. Actually, they're having the language most of the time very comparably to right-handed in the left hemisphere, dominant in the left hemisphere, exactly like right-handed. But you can see that your tail is showing a small distribution of left-handed participants that will have their language completely localized in the right hemisphere, in a mirror of the classical organization of right-handed. So it does happen, but it is extremely rare. So if you see that we say that 10% of the population is made of left-handed people, only 5% of these 10% will show reverse pattern of activation in their brain for language. It does matter because if you have a stroke in the right hemisphere, unfortunately, and you're part of this 5% of the 10%, then you will have an aphasia, which we call a cross-aphasia, which is an aphasia with a lesion in the right hemisphere. It is also important if you want to have surgery, your neurosurgeon must know whether your language is right localized or left localized in the brain. Now, if language is lateralized this way, it probably has some support in terms of the connections that are in the brain. I myself been trying to look for brain asymmetries that will support the functional lateralization of language. And what you can see, of course, part of all the tracks that you can explore in the brain is that the arcuate fasciculus, number 11, if you take it as a whole, is not really left lateralized, but if you focus on the connection that we call the long segment in 14, the connection between the frontal and the temporal lobe, those are extremely left lateralized, whether you look at the volume or you look at the caliber of those connections, of those fasciculi. And why does it matter? And as I said, like, you know, your lateralization for language might be predictive of your recovery after a stroke or the occurrence of your symptom, or might be decisive in the way a neurosurgeon want to take care of your brain, and might also show some different kind of sign of resilience if you are unfortunate enough to develop a neurodegenerative disease. Some preliminary findings from Stephanie Focal, which you will see during the practicals, are already showing that you have a direct correlation between the size of this connection between the frontal and the temporal lobe of the arcuate in the right hemisphere and the recovery for aphasia in patients that had a stroke 6 months before and who were showing language disorder. And this is a nice example where you can see that the brain at the bottom left is a patient that has a very tiny arcuate fasciculus in the right hemisphere. His arcuate fasciculus, unfortunately, is gone in the left hemisphere, and his recovery 6 months after the stroke is actually not good at all because it is below the dashed red line, which is at the top. In the middle, you have another brain with a slightly bigger arcuate fasciculus, and this shows this patient has a better recovery than the one on the left. And finally, on the right, you have this patient that has a very strong arcuate fasciculus. He was showing an aphasia after his stroke, but 6 months after his stroke, he's back to a normal language. And this is really important because he push really forward the idea and the concept that we're not all equal in our organization of the brain, and this has an impact on the trajectory of our recovery. Now, what's interesting is we spoke about lateralization of connection in favor to the left hemisphere and the obvious relationship with language, but you can see that in the arcuate, you also have frontoparietal connections that are right lateralized in the brain and should be supporting specific functions that will be dominant in the right hemisphere. Now you can see these drawings have been done by Wilder Penfield, who was probably a revolutionary neurosurgeon who changed the life and improved the quality of life of so many patients that had a brain tumor. He was the one who introduced the electrical stimulation of the surface of the brain. And so like during the surgery, and you know the brain doesn't have receptor for pain, the neurosurgeon open up your skull and is going to remove the part of your brain that is supposedly being the origin for your epilepsy or being suffering from a brain tumor. So they remove the brain tumor. And they do so because if that's epilepsy and worsen, you die. And if that's a tumor, it grows more and more, squish your brain and you die. So you have, unfortunately, to remove it to improve the life expectancy of the patients. However, like the brain is very complex and neurosurgeon, you know, they've been removing what is not functional, what looks dysfunctional or has to be removed, but it's difficult really to have boundaries onto what you can remove and what you shouldn't remove and the impact you can have onto the functioning of the brain of the patient. So really Penfield brought this revolutionary method, which was to use a small electric current stimulation onto the surface of the brain in order to, in the awake patient, in order to produce temporary 2-3 seconds impairment in the patient, that he was able to map directly onto the brain and avoid damaging these areas that he mentioned being eloquent areas during the surgery. And by doing so, you're able to map the boundary of the speech area, of the motor area, sensory area, visual sensory area, and going so on and so forth into other kind of functions. And then you see it's quite well depicted for his time in the left hemisphere, but when you go in the right hemisphere, there is almost nothing except for this big area in the parietal lobe that he calls space orienting, because patients were reporting disorder when he was removing this area in the patient. They were presenting with disorder of spatial orientation. And this is bringing us to the concept of spatial attention that I usually break down the following way to explain to you the model of how it works in the brain. So spatial attention allows you to select important information around you. So your brain is not omnipotent. It's like definitely limited, as mine is, in the way it can process information around us. So it definitely have to make a selection of what is important in order to guide our behavior. That's a very good system for that. And if you were to be lost, for example, in the jungle here, trying to find your path to civilization, your brain will be able to collect progressively information and cues in order to build up a rational decision on which path you should take. And here you can see the shadows of people on the right path, probably the best way for you to join civilization. This exploration is slow and serial, and you're collecting information and maintaining it to have this illusion that you have this snapshot live stream of your visual environment, but you actually don't have that much information. You're just updating and having this virtual sensation that everything is updated in parallel around you, but it's just like tiny updates that you're doing one by one. This phenomenon is called control goal directed attention. It's typically it's a strategic and voluntary orienting of attention toward visual targets. And if you put people inside the MRI scan and ask them to do this task, they will typically activate a dorsal network involving the frontal eye field and the superior parietal lobule and the intraparietal sulcus. And this is what we call the dorsal attention network, or control goal directed attention. Dorsal attention network being the name that is provided by Professor Corbetta for his model of attention. We'll come to that point. However, life is not always that simple, and sometimes you need to be much quicker and less strategic in order to optimize your chance of survival. So typically in this situation, you got a tiger and your attention will do this. And this is mostly because you have a system in your brain that will bypass your control directed attention. There is no way you can suppress that system. And that's actually the strategies that illusionists are using to make you believe that they're doing magic. They will capture your attention with an event because you cannot suppress it. All your effort and your resources will be dedicated to this event. And they will use this distraction to manipulate something else and make some flowers appear in the left hand. They just took it from somewhere. And you have the feeling that it just appeared like that. Then they just manipulated, tricked your mind by working on this system of bypass of your control directed attention. In that case, this system of exploration is extremely fast and it's data driven and irrepressible. And we call it the grab stimulus driven attention because your attention is grabbed It is an expected and automatic orienting of attention toward visual targets. And you can see that if you're doing this kind of task in an fMRI scan, you will have a different network of activations that involves the ventral frontal cortex and the temporal parietal junction, as well as the DAN. But this ventral part will force the DAN to orient his attention towards this very emotionally and survivally and very important event happening in your environment. So this led us to the wonderful model of Professor Corbetta for the visual spatial attention network, which is divided into a dorsal network, which is controlled, directed attention, strategic and voluntary orienting of attention toward visual target in blue, and the grab stimulus driven attention, unexpected and automatic orienting of attention toward visual target in orange. And those two networks work all the time to orient and filter the information from your environment to optimize your interaction. Now, pretty much, you can see that this has been published in 2002. And when I started my postdoc in 2007, my goal in life was to try to understand how do these activations in the frontal lobe can work with this activation in the parietal lobe. What are the wires supporting it? And indeed, you have some beautiful postmortem dissections that exist from the 19th century, such as the one of Mayo, where you could eventually imagine that some fibers that you can see in the vicinity of the parietal cortex are connecting the frontal cortex, but you know, like there is no real map of it, so it's difficult to know. And on the other hand, if you look at the anatomy of white matter connections derived from axonal tracing in macaque monkey, we have a very clear depiction of how the frontal lobe connects the parietal lobe through three branches of the superior longinal fasciculus, one in cyan, two in dark blue, and three in pink. However, when I started my research in postdoc, all we had really was for frontoparietal connections, the description from Professor Catani of the dorsal arcuate frontoparietal connection linking the ventral to the parietal, the ventral frontal cortex with the ventral parietal cortex and nothing else. And I couldn't think that, you know, the dorsal frontoparietal network, the dorsal attention network will be wireless. So, you know, and I did my postdoc with Professor Catani. The first thing that I did is try to look at all the connections that could go from the frontal to the parietal lobe. And no matter how hard I tried, the only connections that I could see were ventral connections between the ventral frontal cortex and the ventral parietal cortex. At that point, you might be, oh, this is maybe my paper. Is that like there is no dorsal connection at all in the human, and we're very different from monkeys because we have less connections compared to them. And that would explain why we're so bad at spatial compared to monkeys. But you know, those are mathematical reconstructions, and you always need to think whether this is, you're not limited by the method in your observation. A negative finding is very hard to demonstrate. I didn't feel confident in that. And actually, I was right, because if you look at the bottom, if we do like this blue square, and we try to look at the behavior of diffusion in every voxel in this blue square, you will see that so the little smarties, those are like, you know, tensor mole. You've seen in my other lecture how diffusion works. You'll see that they're very round, and they're very round because you have a lot of crossing going in different direction in these areas where you're looking at the connection of the frontoparietal cortex. And the problem with that is, like with diffusion, you just have an average of the orientation of the diffusion of water molecule in that tissue. And because it's a 2 by 2 by 2 millimeters and those connections are just 1 to 5 microns, it is really very likely that you might have different populations. Fortunately, I met the Professor Delacroix, who was like a postdoc like me at that time, who dedicated his life to model really this crossing. And from these little spheres that you can see at the top, we went to these stars that actually is modeling the interaction between different population of fibers in every voxels. By reprojecting that on the entire brain and doing the same analysis of all the connections between the frontal and the parietal lobe, I was able to recover those dorsal connections and show that actually the human brain, very much like the monkey brain, is organized with 3 branches of frontoparietal connections, the SLF1, 2, and 3. As I said to you, those are mathematical reconstruction, and even though I saw what I wanted to see, the real questions that we need to have is whether or not those connections are true. And to do that, you really have to dig into it, and that's what we've been doing. We've been doing some postmortem dissections, old-fashioned style 19th century, well, 1950s, because that's Klingler, where you freeze the brain, and the freezing of the brain is gonna separate bundle of white matter because there is some water between them, just because it's easier to, because, you know, like if you put a bottle of beer inside your freezer, it explodes because the liquid, the water, when it freezes, take more space than when it is liquid. The same idea at the microscopic level in the human brain here. You put it into the freezer and waters can break the organization of the inside of the brain just because it's easier to break along the path of axon rather than across them. Then when you're doing your postmortem dissection, after unfreezing the brain, you can see main white matter structure and peel and see different pathways very clearly. And I highlighted here for you the SLF1, 2, and 3 that we've been able to see on in postmortem dissections. Now that we knew how the frontal lobe is connecting the parietal lobe, we try to explore which of those connections will be supporting the attention system. And essentially, we thought that the SLF3 and the SLF2 seem to colocalize in terms of projection with the projection of the grab stimulus-driven attention. And the SLF1 was projecting onto the system of activation of the intraparietal sulcus and frontal area field and might be supporting the dorsal attention system. Another work from the same group of Professor Corbetta showed that if you compare a little bit like Professor Mazoyer have been doing, the level of activations in the right hemisphere with the level of activation in the left hemisphere during attention tasks, you will have more activation in the right hemisphere than in the left hemisphere, especially for the ventral attention network. And this happens whether you know, whether you can, so you have the caption on the top right, whether you have to detect something happening in the left in black or in the right in red. And the comparison you can see in the chart is the activation for detection in the left hemifield in black and the right hemifield in red. And when it's an empty box, it's the activation in the right hemisphere. When it's a full dark field box, it's activation in the left hemisphere. You can see, as I mentioned to you, that in most of the attention network, you have differences between the left and the right hemisphere activation, okay? This is mostly for inferior parietal lobule and inferior frontal gyrus, middle frontal gyrus, and like the other more dorsal areas were actually not significant. So we try to see whether, you know, this lateralization can be reflected by asymmetry in our white matter connections. And we found that whether you're looking at the volume of the caliber of your white matter tracks, they are bigger for the ventral part of the frontal parietal connections than the dorsal part. So the SLF3 is extremely right lateralized, the SLF2 is slightly lateralized, and the SLF1 is not lateralized at all. And that's a lateralization index that we're doing here. So that's right minus left divided by right plus left. Okay. Now, this is interesting, and this is what we traditionally call converging evidence, but that doesn't mean that those things are statistically linked. So what we try to do is try to quantify how much in terms of behavior people were dominant for the right hemisphere for visuospatial attention network. And to do so, we use a line bisection. It's a long line of 20 centimeters that I'm presenting to you, and I ask you to put a mark in the middle of it. And most of the population of RC participants will miss the center and put their mark slightly deviated towards the left of the true center of the line. And this small left deviation is usually referred to as a pseudo-neglect effect, and is usually regarded as a manifestation of an asymmetrical processing of visuospatial information in the normal brain. And so we try to look at which connection will be related to this effect, and we found that the only connection that is related to this effect is really the SLF2, which is highly correlated with this pseudo-neglect effect. So that brought us to revise a little bit our vision of the contribution of white matter tract to attention, whereby really SLF2 will be mediating the interaction between SLF1 and SLF3 and representing really the substrate, the hub inside the brain supporting this visuospatial attention, filtering the stream of consciousness, visual consciousness that we can have around us. Now, this is about visuospatial, but you might think, you know, how just, is there like a dominance for example, handedness? Because most people are like right-handed, and I'd like to know whether we can find like areas in the brain that will be bigger in the, well, tracts in the brain that will be bigger in the left hemisphere compared to the right hemisphere supporting handedness. Now, we tried to explore this with Dr. Owell, who has been first looking at the lateralization of different tracts in the population, such as a corticospinal tract, which is not so well lateralized at the population level. She looked at cerebellar tract, and the same thing is not so well lateralized, but and she really compared it between the right-handers and the left-handers participants. And when you do that, really, you don't find any difference between the two. However, when you start looking at the frontoparietal networks, you see that you have a significant difference between right-handers and left-handers for the dorsal frontoparietal network and for the ventral frontoparietal network, and not really for the SLF2. So with the same logic, she was, oh, this is interesting. It looks like those tracks support somehow the relationship with handedness. And the way she wanted to assess it was trying to be more than just doing a question, like, you know, just asking questions to the participants, but what is his favorite hand? She wanted to have a dimensional approach to it. So we bought the groove board pegboard and use it to estimate how fast people will be to manipulate small pins that they have to put inside this box as fast as possible with their right hand and with their left hand. And in parallel, we program a little test to measure how fast they will be to tap with their right hand or with their left hand with one finger as their index, or if they had to do sequences with multiple fingers. Okay. And so what we could see, and this is a chart at the bottom, is that you have a direct relationship between the lateralization of the dorsal frontoparietal network SLF1 and how fast you will be to perform, while the difference of speed you will have to perform with your right hand, with your left hand for the groove pegboard, whether you have to put your pin inside the little holes or remove them as fast as possible. If you do a lateralization index, the bigger your SLF1 in the right hemisphere, the faster you will be with the left. The bigger in the left hemisphere, the faster you will be in the right. And what is interesting is like she extended that to as a finger tapping, and there is a slight relationship with how fast you can finger tap with your index, but the sequences is not correlating at all. So this suggests the fact that there is a stronger relationship as you interacting with space suggests that really handedness and the way you can measure the relationship between handedness and white matter tracks is not about really preferring a hand over another, but it's more like the interaction between a hand and space, which will require frontoparietal connections, that is important to determine which hand you will prefer in life to use to do different actions. And then so that brought us to, so in the end, what will be the model of the functioning of the brain in terms of the way you can distribute information between the two hemispheres. If we listen to Roger Sperry, we get back into our localizationist approach where the brain is made of a mosaic of functional regions that are separated and distributed across different hemispheres. And you're going to have your little area of speech writing, or speech, the little area of writing, the area of main language in the left hemisphere, but not in the right. And that's why it has the effect you can observe. If you listen to Marcel Kinspoon, he's working a lot of visual neglect. The two hemispheres work together, and they work together through an inhibition effect, whereby the right hemisphere inhibits the left hemisphere when it needs to work, and the left hemisphere inhibits the right hemisphere when it needs to work. And that's this balance that is important, and that's why when two parties, when you disconnect the two hemispheres, the participant will show this conflictual action. It's just because the left hemisphere is not here anymore to inhibit the right hemisphere action when it's carry on something, and that leads to a conflict into the behavior between the two hemispheres. If that is true, then we will expect that the areas that are the most lateralized for function should inhibit more the contralateral homotopic area, or should inhibit more the other hemisphere, and then we should have bigger corpus callosum connections for the areas that are more functionally lateralized. On the other hand, you have the model of Mike Gazzaniga, which is more supporting a specialization of the hemisphere in interaction with the other hemisphere, whereby really an area specialized for a function to take over all the work and relieve the other hemisphere from all the other functions that it might have. So there is more a complementary action. And if that's true, and that's a relief, then we should expect having less corpus callosum connection with the other hemisphere. So to do so, we try to answer this question by exploring functional lateralization and its relationship with the strength of connection of the corpus callosum. And to do so, the first thing we had to do is create a map of functional lateralization that is as comprehensive as possible. So we collected all the map and manually cured all the map from Neurosynth that I mentioned to you several times, which is a website that collect all the functional MRI activation and create meta-analytic maps, which are, you know, solid report of the areas that are functionally activated during specific tasks. And then we did some important mathematical manipulation on it in order to identify and produce lateralization maps that will be representative of whether we have more functional activation in one hemisphere compared to the other. And so we applied it to 550 functional MRI maps. Before we move forward, we checked whether what we found was true. And in order to do so, we look at the finger tapping. We look at the asymmetry in finger tapping for participants that were doing finger tapping with their right hand and finger tapping with their left hand, and you average the activation. And then you look at whether you typically activate more your right hemisphere or your left hemisphere. So in A, you have the maps that we've been able to reproduce from Neurosynth by doing this meta-analytic asymmetry map. And you see that you have more activation in the left hemisphere at the level of the pre-SMA and the posterior part of the thalamus. And in B, you have true data. This is true fMRI that has been calculated. The patient doing the same task, individual subject, average map, you do a subtraction. You see that you can find an asymmetry in the same pre-SMA area and the same thalamus area in the left hemisphere, plus extra regions that we couldn't see just because we're not as precise as if we had, like in this present case, 200 subjects very finely measured for behavior. Here, this is a meta-analysis, but at least the areas that we report, we know they are significantly and at the top of the differences. And this is the validation of our methods. And then that gave us 550 lateralization map that we try to organize according to whether or not they look alike. And if they look alike, we put them close together. If they don't look alike, we put them distant from each other. And by doing so, we've been able to represent a three-dimensional space whereby you have three tendencies. You have functions that lateralize in that co-lateralize in the same hemisphere for language and visual form and all these symbolic communications. Other functions that co-lateralize and are very similar to each other for the manipulation of emotion in pink, and other functions that have the same pattern of lateralization and similar to each other for everything that is related to the perception and action. And so that gives us like this beautiful triangle. By exploring a little more the data, we found that there was like a fourth dimension. And this fourth dimension colocalized together functions that were showing similar lateralization, but for everything that is related to decision making and reward, which is also a lateralized function. So we ended up with those 4 maps representing essentially the level of functional lateralization of the brain in 4 dimensions. And so you cannot give a single map of functional lateralization in the brain because the lateralization will be different according to different functions. And we've been able to reveal that you have 4 main domains of function that are supporting lateralization. So symbolic communication, within blue, you can see the typical areas of language production. The perception action, with in orange the typical areas that we found activated during the spatial attention, and in blue the somatosensory cortex in the left hemisphere. The manipulation of emotion with the ventral frontal-parietal network that we can see here, some involvement of the amygdala, and the decision making that is strongly right lateralized for the prefrontal cortex, but also involve the intraparietal sulcus. And so those are the 4 dimensions of brain lateralization as we know it. And the next question was, how do this level of lateralization of brain function relate to the strength of connection with the corpus callosum? So we took the best data that we have for the measurement of the corpus callosum connections, and we extracted an average level of connectivity that you can see here at the bottom, that we contrasted or we compared to what we know about how the brain, the left hemisphere, is connected with the right hemisphere in monkeys. And we see that we have some similarities with a ventral part of the somatosensory cortex, which is poorly connected, posterior part of the inferior frontal gyrus, which is poorly connected, orbital frontal cortex that is poorly connected, anterior temporal lobe that is poorly connected, and some stronger connection somehow at the level of the axial gyrus, so planum temporally, that is also comparable to what we can see in monkeys. And then, so we picked up we sample randomly multiple times thousands of voxels from our functional map, look at how they were lateralized, and compare them to how much the connection, compare them to the strength of connection between the left and the right hemisphere. For that, we use an index which is called axon water fraction, which is how many axons do you have really in the midsection of the corpus callosum. And so if you do so, for the left and the right hemisphere, you can see that lateralized lateralized voxels in terms of function are actually systematically less connected than non-lateralized voxels. So if a voxel is lateralized in terms of function, it will be less connected by the corpus callosum than it is if it is not lateralized. We reproduce this effect in the left and in the right hemisphere, as you can see here. Then we try to look at the number of streamlines, and with you know, because there is a lot of crossing, it's a little bit of kind of a sketchy approach to do. And if you do that, you don't have more streamline, you don't have more connection, but they're more densely packed. Okay, but streamline is really not the best connection for this. And then we try to assess whether this number of streamline, however, within the areas that are functionally lateralized were really showing higher probability according to the level of lateralization. And we've been glad to demonstrate that areas with strong functional lateralization have the tendency to demonstrate a smaller number of streamline when compared to areas with a small level of functional lateralization. So, and you might well ask the question, how did we have this relationship within the areas that are lateralized, but no difference between lateralized and non-lateralized for the number of streamlines? But that might well be because the differences between lateralized and non-lateralized areas are a microscopic level in terms of axon water fraction, and within lateralized areas you have these slight differences that exist, which is at the macroscopic level. So in sum, when you lateralize your brain, those areas have been developing in isolation and reducing their connections with the other hemisphere. And you might ask, like, why is that in terms of evolution? Well, it's probably because as the brain with time became bigger, like the communication between the hemisphere became longer because you have to travel further. And in order to optimize like the time of processing in the brain, function get lateralized, and the amount of, and that will reduce the amount of back and forth between the hemisphere that is required in order to integrate information. Moving on evolution, you remember like the variability maps that I talked to you too, that appear to have a relationship with the trends of evolution. So the more, and if you get read the, you get to watch a lecture on brain evolution to follow this path really. But in sum, we demonstrated that the more variable is the brain, the areas that are the most variable in the brain are the latest that appeared on the evolutionary tree. And so this has been confirmed not only because a model of evolution that we can see for the dual origin theory by Sanides in the middle match visually the areas that are the least and the most variable on the right, but also because we have a strong correlation. Well, we have a trend of correlation between in the frontal, the temporal, and the limbic lobe between the level of variability in the human brain and the evolutionary expansion if you had to stretch a monkey brain to make it a human brain. And you have this correlation between the two, which suggests really that you have a significant relationship between expansion, evolutionary expansion, and variability. So an additional step that we investigated was to explore whether you had asymmetries between the left and the right hemisphere in the level of variability that can be reported. And by doing so, we were pleased to see that in the left hemisphere, the superior and the middle temporal gyri are strongly more variable in the left hemisphere compared to the right hemisphere, which suggests that you have recent trend of evolution that happen in there. And in the right hemisphere, you have some change that happen in the paracingulate and the lingual gyri, which might be related to the way we developed really our social relationship in the society, and those are recent evolution as well. In comparison, in monkeys, there is no stronger left variability in the macaque brain, but some areas are more variable in the right hemisphere than in the left hemisphere, particularly in the parietal lobe, where the supramarginal and the angular gyrus are more right lateralized in terms of variability compared to the left hemisphere. And this will suggest that you have some recent trend of evolution that happened in there, or it might be related to the spatial perception of the monkey or the social brain of the monkey, knowing that the macaques that are the more likely to participate to experiment and the nicest are the ones that will be preferentially selected for reproduction by zoos and by laboratories. This might explain why you have most of the variability in there through this manipulated evolution. That's it for today. I'll be pleased to record and provide you with my lecture on brain gradients next week. I want to insist to invite us to the Neurochino every Monday morning. You need to learn how to read and screen papers and increase your knowledge of the current literature if you want to become competitive researchers. And I'll finish with a small word onto the essay. As I explained to you several times, the essay is very important in the overall evaluation of your performance to this module, but that's not the only way you will have your final grade, because your final grade and your evaluation is coming from the interaction between your participation, your attendance, and this essay. Your attendance is completely related to the practical sessions. Your participation will be measured as whether or not you ask questions on the YouTube channel or on Eurostar if you don't want to put your email on the YouTube channel. And this will be summed up to your score at the essay. For the essay, you need to choose a brain hierarchy that you want to write about. You free on the brain hierarchies that you want to write about. But I want your essay to be a total of 1,500 to 2,500 words, not including the references at the end of your essay. Your essay will be divided in 5 sections: a general introduction about what is so exciting about the brain hierarchies that you chose to write about. Get me excited. Anybody who reads this part should be excited, even somebody who's not interested or not a specialist in the brain. Then you need to get into the anatomy of this hierarchy. What are we exactly talking about? What are the areas, the component of this hierarchy? Then a little section onto whether or not this hierarchy is lateralized and how would you measure this lateralization if no lateralization have been reported before. I need a little section about brain evolution in there, where you link up the anatomy, the component, and the lateralization with evolutionary trends. And finally, a good synthesis whereby you wrap this up all together and show me how it all makes sense together. The deadline for this is December the 11th, midnight, and you need to send it to me at my personal address, michel.thibault@gmail.com. I thank you very much for your attention, and I look forward to seeing you during the practical sessions.